I'm now going to model several membrane transport mechanisms. This is very similar to the basic cell structure lesson that I did earlier, where I showed you a number of different things using the same pieces of the model. So the first thing I would like to model is the sodium-potassium pump. Now obviously the key molecule that's missing here is ATP, and again you can come up with your own creative way of presenting that. What our students are taught is that when the pump gets phosphorylated, that phosphate results in a shape change of the pump, and that shape change opens a gap where 3 sodium ions are pumped out, and that shape change also opens a gap where 2 potassium ions are pumped in, and that is a result from 1 ATP being dephosphorylated. Now this is particularly a lesson that's going to be taught at the grade 11 level first, and I usually tell them that that's going to be used a little bit more when we get into grade 12 biology. Now for diffusion and facilitated diffusion, here's a glucose protein channel, and you can see from your legend here's the protein carrier. Now you can see I've done something different here. I've just left an actual gap here that's just the blackboard, which allows this manipulation to take place a whole lot easier than if I had this protein carrier on the membrane itself. So that's really the purpose of me removing that part. The way we demonstrate this to students is glucose is going to come in, it's going to bond to that protein carrier. We do a little dance move, and then we do a spin move where that protein carrier spins and brings in the glucose and then flips back into its original position. So that's a really clear model of facilitated diffusion. Now just diffusion itself, if you can see what I did here in my setup a bit, I bent this membrane a bit so there's a little bit of a gap, and here we have high levels of oxygen, and this is demonstrating now that oxygen can come right through the bilayer. And it probably just hit a magnet there. I'm going to lift that up a bit and pull the oxygen through. So again, I'm trying to communicate to the students that certain parts or ions or in this case molecules need to go through those protein channels. Oxygen can come right through the bilayer. Now I know you're all waiting for this endocytosis move, so you can see what I've done here differently. There's 1, 2, 3, 4 curved molecules. 4 curved cell membrane pieces, I should say. In this particular case, a molecule that's going to bind to that membrane surface is going to take parts of the membrane with it, and these are going to fuse together to create that vesicle, and that's why we have that vesicle formation as part of your lesson. And the vesicle is carrying that, in this case, protein in a protective cap. The other thing I like to tell my students now is that the membrane has a gap in it, and to demonstrate the fusion of the remaining membrane, I take those pieces and make the cell membrane smaller, and it's clear to the students why the cell membrane is smaller and that this vesicle was actually part of the cell membrane. That helps the students understand, like viruses budding out in exocytosis, for example. So you could just do the reverse move and exocytose that vesicle, and that would demonstrate that clearly too. To conclude the membrane transport lessons, you can see that I've built a different picture here to show you the function of insulin. So I've just built a smaller cell here. Inside the cell there's a vesicle that contains a stored glucose channel. Here we have a glucose channel in the membrane, and here is an insulin receptor. Now up here, this is insulin. This phospholipid bilayer is representing one pancreas cell that contains insulin. If we look at the receptor now, if I separate the inside of it, you can see that I've used these two pieces to make a larger tail on the inside, and that's going to be a shape change, and you'll see the result of that in a minute. So the way I teach this lesson to the students, I mean, again, they would take a note on this. It's not really a classic before, after, or during lesson, but they would draw this picture, and then again, it's up to the student to add notes to the pictures as I start to teach, because it's a fairly short lesson, and they can use words and pictures. So the students have been taught about, glucose coming in, and this particular lesson is, okay, we need here now to have a certain volume of glucose in the blood all the time. So if you eat and increase your glucose levels, this, I tell my students, is like the temperature going up. So what is the mechanism of the body reducing the glucose levels in the blood and getting them back down to, let's call 3 normal. So that's what this lesson is about, really. How do you lower blood sugar levels when they're high? So tell the students that there's interesting mechanisms, and again, like all the lessons, you can go into whatever kind of detail you want. The pancreas gets messages and is going to release insulin. Insulin is going to bind to insulin receptors on cells. That causes a shape change inside the cell, which is going to end up having that vesicle be transported to the membrane and quickly deliver that extra glucose channel. The vesicle can return to shape. And now this cell, not to mention, trillions of other cells, are going to have the mechanism of taking in now twice as many glucose as they did before. And now the students can see a mechanism of returning glucose levels back to normal. This can lead into an interesting extension, which is the reverse of this lesson. You see I've removed 2 glucoses. So now we have low blood glucose levels. And again, that's low in my calibration of 3 being normal. So I'm going to throw out a challenge to you there and the challenge is take the pieces of the protein membrane kit that you have and start to play around and create a lesson where you're going to introduce glucagon and you're going to raise these blood sugar levels. And I would love to hear what the result of that lesson was. You can contact me through my webpage. And I'd also like to hear any other feedback on any of the lessons that you've seen me demonstrate, and even more in particular, your feedback on what it was like to teach with the model. So I would be very interested in reading all those comments. And I would just like to thank you for participating.